Hi, I'm Hitoshi Okamoto at Brain Science Institute of Riken. Making the right behavioral program to avoid dangers and choosing the most suitable one among them under a given situation is the essential capacity of animals for their survival. Elucidating the neural mechanisms for this process is one of the most important issues in neuroscience. One promising approach to address this question is to use the model animal with a brain which is enough simple to study and yet still conserved functionally and structurally with mammalian brains. The teleost fish brains used to be thought to have completely different structures from the mammalian brain. However, the recent study has revealed that their telencephalons are by far more similar to the mammalian telencephalons than people used to think before. This knowledge has made the results obtained in fish much more interpretable from the common perspective of the vertebrate brain, and has made the fish an excellent model animal to study the neural mechanism of adaptive behavior.
Hi, I'm Aoki, a postdoc in the lab of Hitoshi Okamoto. By imaging neural activity across the whole telencephalon of zebrafish during the learning and memory of an avoidance behavior, we aimed to visualize how the collective behavior program is encoded and retrieved during decision-making. To achieve this goal, we established an active avoidance learning chamber for zebrafish. In this chamber, fish that have been genetically modified to express a genetically encoded calcium indicator are trained to associate a red LED light with an aversive mild electric shock by swimming to the opposite compartment when the red LED light turns on. Thus, after learning was established, fish quickly go to the opposite compartment to avoid the mild electric shock upon the presentation of the red LED lamp. Once the fish learn this avoidance behavior, we then place them under this imaging setup to visualize neural activity across their entire telencephalon as calcium signals from the origin. We present the red LED light as a stimulus for avoidance behavior that the fish acquired in the active avoidance paradigm. Untrained control fish to which only the red LED is presented showed no activity in their telencephalon upon the presentation of the red LED lamp. In contrast, fish which learned the avoidance behavior showed highly localized bilateral activity in their telencephalon when examined 24 hours after learning. However, 30 minutes after learning the avoidance behavior, neural activity was not observed. This indicates that the highly localized neural activity we observed is specific for long-term memory and memory retrieval. We then examined whether the cortical regions we identified by imaging are necessary for short-term learning and long-term memory. The ablation of these regions in the telencephalon did not affect learning or memory retrieval of the avoidance behavior at 30 minutes after learning, whereas memory retrieval 24 hours after learning was impaired. This result demonstrates that a long-term behavior avoidance program is encoded in the region we identified by imaging. We next asked whether and how a change in the learning rule of the behavior paradigm modifies neural activity patterns in the telencephalon. For this purpose, we trained fish using two different cues, either a red or blue LED light, where each color of light is associated with a different learning rule. In this case, red light means avoiding the shock by changing chambers, and blue means avoiding the shock by staying in the same chamber. We then analyzed neural activity by calcium imaging upon presentation of the red and blue cues, and found that neural activity in the two different behavioral rules differ from each other. This result indicates that the rule change recruits an additional set of neurons to control a different behavior.
In this study, we succeeded in imaging the activity of neural ensembles that are activated when a behavioral program is being retrieved in a part of the zebrafish brain, which is likely to correspond to the mammalian cortex. In the near future, taking advantage of the small size of the zebrafish brain, we will be able to observe the interaction of these activated neurons with deeper areas of the telencephalon, such as the basal ganglia. Making the right choice of a behavioral program under an incessantly changing environment is essential in our daily life. And the inability to perform this maneuver underlies various psychiatric disorders. Further study on this process using the zebrafish system will contribute to the elucidation of how this process works normally and what can happen when it goes awry.